The way I want to start is by saying that this movie that you're watching in the back of this introductory slide is actually a zoomed in image of a biological tissue. And I'm not going to tell you too much about it now because we'll talk about it again later, but I want you to notice that the white outlines are the outlines of cells, and you can see they're sort of jiggling around and moving a little bit. And we deeply want to understand this, and I'll try to say more about why. Okay, so this is an interactive talk. So the first question is are you a solid or a fluid? I heard both solid and fluid. That's good, because that means that you can chat with your neighbor and see if you can figure out what the right answer is. No, I think both answers are kind of right. So somebody who said fluid, why? You're made up of a lot of water, right? And that's completely true. What about somebody who said solid? Why'd you say solid? Rigid, right? Because as a vertebrate, as an animal, you have to move around. You have to support shear stresses and walk. And in order to do those things, you have to have a solid-like behavior. And, you know, if you pinch yourself, what you usually think is your corporal body, right, it sort of relaxes back. It hurts a little bit, and there might be a little plastic deformation, but it's a solid, okay? But I want you now to think for a second about what might have happened earlier on in your life, very early on in your life. And so this picture that I'm about to play, I have to use this analog pointer, sorry. So this here is a zebrafish embryo. And what you're going to see when I press play, this is the yolk of the embryo. And the thing that's going to become the animal starts from those cells up here. So let me play the movie. You see it divides and divides. And then it begins to flow around the sides of the body. And this stage is the gastrulation stage. And so you might think, well, during this stage, when you have very large motions, really it might behave a lot more like a fluid. And if you zoom in, these are some beautiful movies taken by a collaborator of mine, Eva Maria Schatz-Collins. And these are nuclei. So this is a very zoomed in picture, even more zoomed in than this. And what you can see is that in the same process, sort of here, you're now watching the individual nuclei move around and jiggle around. And I think really to your eye, it probably looks a lot like a fluid as well. Okay, and so I sort of want to make the first comment that tissue fluidity is actually really important during development because it allows the cells to go to the right places they need to be. For example, there needs to be a segregation of tissue types. The cells that are going to become your brain have to get segregated for the cells that are going to become your gut, and you actually need these large-scale flows in order to get that segregation. So this is over the course of about a day. So this is over one day. This is a much shorter time scale. This movie is about 4 hours. Okay? Yeah, so these are very sped up. Great question. Okay. So the other thing I want to say, can you raise your hand if you think of yourself as a scientist in this audience? Okay? How about not a scientist? Great. So we have a good mix. Okay. And so I wanted to point out that for those of you who don't consider yourself scientists, whenever you see something in blue like this means it's a scientific paper, and that means it's been peer reviewed by your peers or the scientific peers. And so that's what that little thing means. Okay. So another time when you might think about fluidization is when something goes wrong. Okay. And so normally, when things are going right, a lot of the parts of your body are solid-like, okay? But when you have a dangerous cancer tumor, what has to happen is the cells have to leave the primary tumor and move and flow through the body to get to a new place to start a new tumor. And so you can actually see that this is what's called a breast cancer cell line. So these are different types of cells. This is a model for normal cells. This is one for malignant cells, which are bad, but not really, really bad. And this is one for metastatic cells. And then what some scientists, Joseph Case's group at Leipzig University has done is to take balls of cells. So each one of these is a ball of thousands of cells, and they've put them in a petri dish and imaged them over time. And here I've actually listed the time so you can get an idea of the time scales involved. And when I press play, I want you to focus on this one first. So what you can see is those balls of cells fuse together and round up. And actually, that's exactly the functional form that you would see if you watched oil droplets round up in water. to immiscible fluids. And then over here, these metastatic cells actually really barely fuse at all over the time course of this experiment. And then there's this sort of interesting behavior, and this is very robustly reproducible, where these things sort of partially fuse but don't all the way fuse together, and they become arrested in a characteristic shape. And so what I would like to suggest to you is this one is behaving very much like a fluid. This one's really behaving mostly like a solid, and this one is something weirdly in between. And so this is another example of where you might think of there being sort of a transition between a solid-like and a fluid-like behavior in these systems. Finally, another example which is very industrially important is wound healing. And so, I could never be a doctor. These pictures gross me out. But if you do experiments on cells and you try to understand how they sort of move to close a wound, you get images that look like this. So each one of these things highlighted here is a cell. So again, this is under a microscope. And you can see that sort of if you have sort of initially a strip of cells, and it's the right cell type, it will flow outward in order to sort of explore space and heal a wound. Okay, and so we would like to understand these processes. Another example, because I think it's particularly interesting, it sort of was surprising to me, is folks with asthma. And so one of the things that you can do, it turns out, is take lungs from the cells of human patients, and you can culture them in an air-liquid interface, and then you can watch them over long periods of time. And so this is a bird's eye view of cells, and so you sort of are looking down on a sort of two-dimensional layer of cells, and these are the number of days after which they've been taken out of a human patient. Okay, so this is 3 days afterward, 6 days afterward, 10 days afterward. And what do you notice about the progression of motion here? Right? Yeah, they're slowing down a lot. Here they're moving a ton, and then less, and then even less. And it turns out actually that you can look very precisely at the motions of these cells, and you can make maps using color of how fast the cells are moving to quantify this. Okay, and that looks something like this. So this is data from non-asthma patients, and the color bar tells you how fast the cells are moving. And so you can see cells are moving a lot here, but by day 6, they're hardly moving at all. Make sense? And then from asthma patients, something very different happens. There's a really big change in the motion. So at day 6, when the normal, sort of the non-asthma patients are sort of solidified, on day 6 for asthma patients, they're still moving around a lot, and they're even still moving around a lot at day 10. And so this is an indication that sort of one way of understanding some of the pathologies of people with asthma is that the cells really don't mature their contacts very well, and they sort of have a delayed fluid to solid transition. And so we'd like to understand that too.
Why are they moving in the first place if you're healthy?
That's a great question, and one that's not deeply understood. So actually, the idea is that when you have a bronchospasm, so like in any, like if you have a coughing fit or something like this, they can activate this type of behavior in these trans wells up there by applying a big pressure to the system. And so these cells are activated by pressure and stresses, and then they sort of want to crawl along one another. And there's some biochemical signaling pathways involved. I won't get into all the details. But actually, under pressure, this cell type likes to move around. It gets activated. Okay? And so we think that perhaps when you have a bronchospasm and they don't quiet down very well, that's one of the pathologies of asthma. Okay. So how do you tell the difference between a fluid and a solid? Okay, I give you a black box, and you trust me, so you stick your hand in the black box, and I say, how do you tell if the thing is a solid or a fluid? Okay, so if it's a fluid, if you push on it, what happens? It sort of moves with you, right? And if you think of sort of, it moves to fill space, right? If there's extra space, you can pour it out of a cup. And also if you think about sort of the force that resists you, when you're, is it harder to sort of push slowly or quickly through a fluid? Right? It's harder. It's like running through a pool, right? Like if you're in a pool, it's much harder to run than walk. And that's because if you're a fluid, how hard it is to push on it is higher as you move faster. That's sort of the defining characteristic of a fluid. Okay? It doesn't return to where it started. And then if you look at the microscopic scale, the particles that make up a fluid, or the atoms, molecules, they rearrange and they change their neighbors. This is sort of how to think about a fluid if you had a black box. Okay, and now let's think of, like, my favorite example of a solid is a rubber band, because there's a lot of them around. And so now when you apply a force, right, you stretch it out, right, how hard is the force? Like, what does the force in a rubber band relate to? Yeah, how far you've stretched it, right? We're all familiar. So the force in this case, how hard it is to push on it, is higher as you move farther, right? So for a fluid, it matters how fast you move. For a solid, it moves how far you move. Okay? It does return to where it started, and also the constituent particles do not change their numbers. And these ideas are intimately related. Okay, and so, all of you are familiar with this today. How do you change a fluid like rain into a solid like snow? You change the temperature, right? Okay, and so when you change the temperature and you cool something down, there's a lot of materials such as water that crystallize when they're cooled. Okay, most metals are like that as well. And just to say, this is sort of a macroscopic shape, and then if you can zoom in a lot, you see sort of an ordered microscopic structure, nanoscopic structure. But there's a lot of materials that don't crystallize when they're cooled. And they're around you, and they're industrially important. Plastics, glasses, fiber optics, these things are really interesting. They used to be in some iPhones. And when you look at the structure on the smallest scales, again, this is the zoomed in structure, you can see that it's really disordered. Okay, so these are what I mean when I'm going to refer to glassy materials for the rest of this talk. And before I go on, we just talked about how temperature could change from a fluid to a solid, but are there other ways? So there's some ringers in the group that are physicists, right? How about sand? Okay, so is sand a fluid or a solid? So you can stand on it at the beach, right? It's supporting your weight. It's clearly a solid. Okay, but the instant you pick it up, this is dry sand, it sifts through your fingers and it looks very similar to a fluid, right? And so you could ask, what's the difference between those two conditions when I was standing on it, when it was sifting through my fingers, right? It's a change in density, okay? And so there's another way you can get to a fluid solid, changing the pressure, changing the packing fraction or the density. My favorite example, because I love mayonnaise, is another example. So how many of you have made mayonnaise before? Yes. This audience is awesome. Usually I don't get that many people that make homemade mayonnaise. But it's interesting, right? You take two fluids. You take oil and you take water and then maybe a little egg as a surfactant, and you mix them together and somehow you get something that keeps a peak. And the fact that it keeps a peak tells you that somehow it's turned into a solid, right? Okay, and so how does that happen? Well, it happens because you keep adding more and more oil droplets in until you get a dense enough packing of oil droplets that they're so tightly packed, just like that tightly packed sand, that they solidify. Okay, and so the summary, and this is what it might look like on the inside if it's mayonnaise, and there are many other structures, sort of materials that have these types of properties. I showed a few. But the summary is that the fluidity depends on the temperature, which you can think of as the energy available for the constituent objects to move around, and the density, how tightly packed the objects are. Okay, and so the big question that there's a glass collaboration that I'm a part of as part of the Simons Foundation, and the big problem, there is trying to understand how these disordered materials become solid, because for crystals there's sort of deep reasons why breaking the symmetry and having this crystalline structure leads to rigidification, but there's no similar understanding in disordered materials. And so to try to sort of unify some of these ideas, this was a sort of an older picture now, almost 10 years ago, of how you might unify sort of changes to density. So this axis is changes to density, and this axis here is changes to temperature. So if I go down that way, this sort of one way of thinking about it is if I cool the system and get a solid, you can sort of call that something like a glass transition. That's the terminology. And then if you go along this axis sort of at zero temperature and you change the density like sand particles, then you get something that looks like a jamming transition. And actually, sort of the behavior, this is an older plot, and so actually a lot of the folks in the collaboration are trying to understand that, there's a new understanding that some of these sort of things in this picture need to be modified. And then I just want to point out, because I think it's really cool, that there's actually another axis that the collaboration is exploring here, which you might not expect. perfect. Okay, and so the other axis that this collaboration is exploring is this axis, which is 1 over the dimension. So here's our old friend, the temperature, okay? And here's a weird axis, 1 over dimension. What does that mean, okay? And so actually, if you look at this axis, there's 1 over the dimension equals 0, so it's like infinite dimensional systems, right? That's what this corner means over here, and that might seem like it might be hard to wrap your brain around, but thinking about high dimensional systems is not so tricky, right? Like, let's say you're looking for an apartment in New York City, right? And one axis is like the number of windows that's in the apartment, right? You want to sort of optimize the number of windows in the apartment. Another axis is the square footage, right? And another axis is how many bathrooms it has. And you can see you could keep adding dimensions to this idea in your head of what would be good for an apartment. And so when we're talking about these things, I think there's sometimes an idea that high dimensions are mysterious, and they're really something that we all think about all the time. Okay, and so what this collaboration is trying to do is solve, there's some beautiful mathematics that can be done to solve what's happening on this sort of infinite dimensional line, and we're trying to extend it further. Just to give you a picture of some of the cool theory things that are going on. And some of the questions that people are trying to answer are questions like this. Is there a most dense glass? How does the stiffness of the glass depend on things like temperature and dimension and strain and things like this we just talked about? And how does the motion of particles and atoms depend on these parameters? Another important question is, how does the glass fail? Because a lot of these materials are structurally important, right? And so I think it's amazing, we don't have a good theory for how disordered solids fail. The only way you can decide how strong one of these things is to do an experiment, right? Every time, in a new geometry, you have to do an experiment every time. So it's kind of amazing, okay? And we'd like to understand the relationship between these disordered systems and lots of other things that are very active, things like machine learning and encryption right now. There's a lot of relationships there. Yes? Okay, so you're exactly right. There's a whole class of materials that are sort of viscoelastic materials that you can think of as somewhere between being a solid and being a fluid, right? And so the reason that it's interesting to try to delineate when something becomes sort of like using a sort of a line to say when something becomes a solid or a fluid. The technical definition is that it has a shear modulus, and what that means is if you do this to it, there's a restoring force. And if you keep pushing on it, you can get all sorts of responses on long sort of different time scales, but it's really useful to classify things in this way because it really tells you about the long time scale limit of the problem. And we find that, you're right that there's all sorts of behaviors in between, but characterizing the long time behavior is a really useful way of trying to understand failure, for example, in these materials. Okay, so I promised you to talk about biology, right? And so far I haven't done that, and you came here in the snow, right? Okay, so I need to tell you about biology. And in particular, you're gonna tell me that, well, of course, cells can do many, many things that mayonnaise cannot do. They divide, they die, they differentiate, which means they turn into different cell types, they respond to their environment, they move around on their own, right? And so what is important about these ingredients if I want to understand the sort of properties of a biological tissue? And so for the rest of this talk, I will ask what we can explain by adding what I'll call 2 simple ingredients to this glassy picture that are relevant for biological tissues. And I'm going to ask, what can we predict for biological tissues and for our colleagues that are working in biology, and see if we can make progress adding those 2 simple ingredients. Okay, and so cells can exert forces and move on their own. They're active. Okay, and there's an interaction between cells that's really interesting and sort of, I would say, a little bit different from what you see in particulate matter, things like molecules or grains of sand, and I'll get to that in a minute. Okay, so how can we describe cells that generate their own forces? So this is sort of a picture of nuclei standing of cells on this projector. It doesn't show up so well, but we're sort of tracking a bunch of cells. This is an experiment with some collaborators, and I'm just showing you trajectories. Oh, that cell divided, and then it's moving around a little bit more. And so you can see that these cells that are crawling on a substrate have these sort of interesting trajectories. They have a speed with which they move. But the other thing that's important that we find is actually really important is how long does a cell sort of move in a straight line before it sort of veers off to the side? That's a measure of its persistence, okay? And so we can characterize cell trajectories by these 2 parameters. And if a cell trajectory has a small persistence length, it looks something like this. And if it has a long persistence length, it looks something like that, okay? And when you have systems that have both a speed and a persistence, when they're active, when they're actively sort of generating their own energy and living in an environment, you can get all sorts of really cool phenomena that it's called sort of this, there's new types of non-equilibrium physics that emerge. And so you can study things like bird flocks. So this is a picture of a flock of birds that you can see has really, I think, interesting, clear collective behaviors that have emerged from the interactions between these active objects. And you can even do something similar if you're looking at sort of, this is on a very small scale. This is colloids under a microscope. So this is at the scale of microns, very, very small. And you can activate them with light, and they can behave in really interesting ways as well when you can activate them and they can move. Okay, so there's this whole new class of active matter models that does really, really weird and unexpected things. Okay, that's the first ingredient. Okay, and then the other thing that we notice is that cells interact with each other differently than particles or droplets. Okay, so this is a picture, again, inside basically some mayonnaise, and this is a picture of some cells. Okay, and then we can say, you know, these are basically as tightly packed as possible. There's really not any sort of space between those cell-like objects. And, you know, this microscopic structure looks really glassy. It looks like it's at a really high density. It looks like I've packed it really, really tightly together. So a naive expectation would be this should behave like a solid. It's really, really tightly packed. I showed you that axis before. And what you see is this is the video I showed you at the very beginning of the talk. So this is a two-dimensional slice through a three-dimensional aggregate of tissues. This happens to be zebrafish embryo cells. And what you can see is that even though it's really tightly packed, you can see that the cells are jiggling around and changing neighbors. And on long enough time scales, this thing actually behaves like a fluid. You can show that it does. Okay, and so we want a good model for how the cells interact with one another, because that seems to be different from what we might expect. Oh, yes?
Why is that contradictory?
So that's an excellent question. So I'll sort of answer it in two parts. The first thing is that we can sort of first take the limit where there's no temperature, no fluctuations. And in that limit, you're thinking about droplets or grains of sand. And in that case, it's pretty clear that in that limit, you wouldn't expect things to sort of rearrange if they were really tightly packed. Now, in a fluid, as you say, actually, there's sort of an energy potential between two molecules that you can sort of model in various ways. And then there's a density there, but it's not like a hard packing problem. And so you're exactly right that thermal fluctuations in that case, sort of the concept of density is a little less clear. You can still talk about a pressure, but a packing fraction is less important in that case. But I would argue that because these things look a lot like foams, that you might ask, why don't they behave like them? Okay. So if we need a model for how cells interact with each other, then we have to ask the question, what mechanical forces act to regulate cell shapes? And there's been a beautiful amount of work on this by lots of biologists and biophysicists over the years. I'm going to give you a really short summary, which is, first of all, cells have these sticky molecules called adhesion molecules that stick them together. And so what that tends to do is where two cells touch, if there's adhesion molecules together, that tends to want to lengthen the interface where the cells touch, because they get a little energy bonus when they stick together. Okay, and so that's here. And then there's an active cytoskeleton that lives sort of at the periphery of these cells. And that basically acts like a sort of a ring that tries, ring in 2D or sort of a globe in 3D, that tries to keep the cells rounded up into a round shape. Okay? And finally, these cells are filled with fluid, and fluid basically doesn't change its volume very much, and so the cell types that we study don't change their volume easily. And so if you put all of those ingredients together and try to write down a mathematical equation for that, you get something that looks like this, and this is the only equation that I will have in the talk, but it's not too bad, so I want to tell you what it means. What it means is that if I take a top-down view, so I have, again, one of these layers of cells, and I take a top-down view and I look at it, then each cell has a cross-sectional area, which we can call A, and a cross-sectional perimeter that we call P. So you can see that, a perimeter, and then the area is the filled-in part, right? And the idea of this energy functional is it says, well, there is a preferred area, and there's a penalty if my area is different from that area, because I pay an energy penalty if this area is different from this one. Okay, and then I have a perimeter, and then I pay a penalty if the perimeter is different from the preferred perimeter. And you can actually show that all of these sort of ingredients that I told you about before sort of arise naturally in this energy functional. I'm not going to go through the details, but sort of all of those appear in an energy functional like this.
It's moving to reduce the value of that function?
Exactly, it's trying to minimize that function. That's the assumption. Okay, and so you can say, well, how good is this? It's sort of a kind of a crazy assumption, and there's been a lot of work. I think I have it up here, actually some by Sasha in the audience right there, that sort of shows that this is sort of a surprisingly good model for a lot of epithelial tissues. It's sort of been well vetted in things like fruit fly eyes and things like this. Okay, and so what we wanted to do is we wanted to see if we could use it to understand dynamics of cells in these tissues. And so I think if I can go back, I can play this movie. And so this sort of red cell looks like it is crawling through a tissue, doesn't it? Kind of looks like it. Okay, but it's actually, this is not a simulation of that. It's a simulation of just a calculation of a bunch of what I would call T1 transitions. And that's just when I start out with two cells that are initially neighbors, and then the edge between them shrinks to a point, and then it grows in the opposite direction so that I end up with new cells being neighbors. And that's sort of the standard unit that can happen in one of these two-dimensional systems. And so all this picture is that looks like a cell crawling is just a series of executed T1 transitions. It's just a series of this. And one can show that that's actually the only thing that can happen in 2D. Okay, and so then we can measure, well, how much energy is required to do that thing? Because I have to do that thing a lot in order to have a fluid-like behavior where cells are changing neighbors. Okay, and so we measure that energy, and we find something that was really surprising to me, which is that this model predicts that there is a fluid phase and a solid phase. So that model had been around for something like 20 years, right? And there is a fluid phase and a solid phase, and the transition between these two was governed by sort of actually a really simple idea. So if I go back to this energy functional, it's basically you say, okay, I take this preferred perimeter that a cell wants to have, and I divide it by the square root of the area to non-dimensionalize it. And that's the important parameter that decides whether I'm a solid or a fluid. And sort of even more interesting for folks who have done sort of this sort of generic glassy physics, in those disordered glassy solids, it's really rare to be able to look at a picture of a disordered solid and decide whether it's fluid-like or solid-like. There's no really obvious way of looking at a picture and telling which one it is. But for this model, it's really easy. So all you have to do, apparently, is you look at the actual perimeter of a cell, and you look at its actual area, and you find if you measure that it's sort of this perimeter to area ratio is large, so it sort of looks like a pancake, right? So let me say for a second what we're talking about, right? If I have a large perimeter relative to my area, I look like this, right? And if I have a small perimeter relative to my area, I look like this, right? And so we're changing across there. And so it turns out that if it's large enough, and there's a critical value that actually is sort of doesn't depend, it's a priori prediction of the model. It doesn't depend on any model parameters. And so if this thing is greater than 3.81, it's a fluid. And if it's less than 3.81, it's a solid. And that comes out of just thinking about this model. And so you can look at pictures of what happens. So this is what one of these model simulations of a fluid looks like. And this is what a model solid looks like. So here you can see cells are changing neighbors, and here they're not. So that's how you decide whether it's a solid or a fluid. And you can sort of use this mathematics that we've developed and we and others have developed to make a phase diagram that says, okay, on this axis is the shape that a cell wants to have. So if it wants to be more like a pancake, then it's a fluid. That's this region out here. And if it wants to be more rounded up, so not like a pancake, it'll be more like a solid. Or if it wants to exert more motile forces, so this is like your question about temperature in fluids. So if there are sort of more fluctuations, then I can fluidize the system. And actually, I think this one's kind of interesting. You can actually take something which is a solid, like a tissue that's a solid, and turn it into a fluid just by changing the persistence of the cell trajectories, right? So this says that if a cell it's not moving any faster or not exerting any more forces, if it just wants to move in a straight line a little longer before changing its direction, you can actually fluidize the system that way, which is kind of neat. Okay. And so the prediction then is that in here, in the solid-like regime, we have a small shape index, and in the fluid-like regime, we have a large shape index. Okay? And actually, the prediction is that in this solid-like regime, the shape is exactly equal to this number in the whole phase. Okay, so that's a strong prediction, but we had this data from those asthma patients, and it's actually really nice data for us because they can very carefully tell when their system is solid-like or fluid-like. So you can actually, there's a lot of different ways of calculating it, but clearly this is fluid-like and this is solid-like. And so then they can actually segment these images and measure these cell shapes directly. Okay, and so what we would predict is that this one has a shape order parameter that's exactly equal to 3.81 because it's jammed, and this one has one that's, because this one's almost jammed, is basically 3.81, and that the shape parameter rises away from that as you get further away from the fluid-solid transition. And so this is what the data look like. So this is, again, this is the cell shape, and this one is jammed, and this one's almost jammed. And so what we would expect is that if we sort of sit here, we should be 3.81, and it should rise away from that as it becomes unjammed, right? And this is no fit parameters theoretical prediction, and this is what the data looks like for a biological tissue. And so our biology collaborators are really excited about this because it basically means that you can look at a snapshot of cell shapes and understand if a cell can migrate in the tissue or not, which is really exciting for them and can be used as a diagnostic tool. And it's sort of, it was nice because it was a pure theory prediction, and we made it before they measured it, right? So that's kind of neat. Okay, and so I want to talk about that asthma is one application, and I want to talk now about some other potential applications of this type of work. And one application is thinking about establishing left-right symmetry in congenital heart defects. Okay, and so this is, I mean, it's amazing to me, right? Almost all of us end up with our heart on the left side of our body. And it's actually important defects in doing so lead to heart defects, which are one of the sort of largest causes of congenital problems in infants. And so you'd really like to understand this. And so as it turns out, actually, in all vertebrates, the way that your body decides whether there's left from right is there is a ciliated organ. So I'm going to show you a picture of this organ in zebrafish, which is a model vertebrate. And there's this ciliated organ that sets up a fluid flow inside this lumen, which is basically an object filled with fluid, and it has to set it up in exactly the right way, going counterclockwise in a particular sort of view of the system, and then the cells read out the stresses from that fluid, and that sets up left-right patterning, and it's the same way in all vertebrates. Okay, so why are we thinking about this problem? So this is a picture of that zebrafish embryo again. That's the body. And this is this little vesicle, which is a fluid-filled vesicle with these cilia here. And the reason that we're thinking about this problem, or we thought we might have something to say about it, is because you can take a slice through here. If you sort of take a little slice and look at a picture of what the cells look like, they don't spontaneously. They always form this sort of asymmetric structure anterior to posterior, where the cells on the top are really long and skinny, and the cells on the bottom are really short and squat. And so our collaborator sort of across the street has shown that this shape change is necessary to get all of the consequent patterning. In other words, if you break this, then you break everything else. Okay, and so this is because we've been thinking about models for cell shapes. We thought we might have something to say about this, or we're thinking a lot about it. Okay, and so the question is why does it do that? What causes these asymmetric cell shapes? And so one of the things that you can do fairly easily is you can say, well, I don't know what it is, but let me assume that there's some biochemical signaling pathway, something called a morphogen gradient, which is basically sort of biochemical signaling molecules that live in the zebrafish, and there's a spatial distribution of them. And then let's say that the cells can read that out and change, say, the tensions on these interfaces. Okay, and so if they could do that, one can show that you get shape changes that are very similar to this. And you can go in and you can actually knock out molecules that cause tensions, and you can see that if you knock out all tensions, you definitely get this thing, but it's not really definitive. Okay, so one possibility is there's just this big gradient of biochemical signaling pathway that does it. Another possibility is this organ actually lives at the bottom of another structure called the notochord, and there's some interesting interactions between the notochord locally and the cells in here. And so you can imagine that maybe these cells are sort of pooling on these cells below it. These sort of red cells up here are pooling on the green cells, and that might do it too, right? So again, you're sort of saying that there's something else that sets up the gradient, but basically this notochord, and once it's there, it can do the shape change. Another thing that you could look at is the changes in cell volume. And so you see, again, it would have to be driven by some morphogen gradient, some signaling process I don't understand. But once I set it up, I could change the volume, and we can show that if you change the volume of cells, that can generate the shape change. But the interesting thing I want to tell you about today is that it could be just physical forces, really, without any need for any sort of gradient in anything. Because as this organ develops, so this is a picture of the cells in this organ, but this organ is in the middle of a dense tissue. There's a bunch of cells surrounding it, and tail bud cells. And so as this organ is developing, right at the critical stage where it changes sort of the cell shapes, it actually is pushing through this tail bud tissue. Right? It's sort of moving through. Okay, and so if you think about it from a fluid dynamics perspective, right, basically what would happen if you took a cylinder and you pushed it through some fluid, right? You would get some forces on the front of this object that sort of squish it, and maybe you'd get some forces that extend it in the back, right? And that's actually the cell shape changes that you see. And so what we can do in one of these models is we can actually say, okay, let's just take a model of this lumen, so that's a picture, an idealized model of it, and ask what happens if we just push it through the tissue. Can we get shape, you might think that maybe you could get shape changes that look a little bit like what you see in the experiment. And in fact, you do. So you push it through a tissue, and you see that actually these cells on the top get really long and skinny, and the ones get short and squat. And there's nothing mechanically different about the cells on the top and the bottom. It's just a result of the forces of the tail bud cells surrounding the organ. And so you can actually do an experiment where you measure the speed of this KV through the tissue, because this effect, of course, depends on how fast you push it. So the question is in real life, is it going fast enough to generate reasonable forces to generate these shape changes? Okay, and so you can do the experiment, you can measure how fast it's going, and what we find is that when you do all of the sort of matching between the experiments and the simulations, these are the possible choices that are sort of work well with the experimental velocities. And so this here is a measure of the cell shape asymmetry, and these colors here are basically right here, it's sort of 0.8 or 0.9 is what you see experimentally. So what this plot is showing you is that exactly in the regime where you actually see velocities that are what we see in experiments, you see cell shape changes that are basically identical to the shape changes you see in the embryo. Right? And so this is I should say to a biologist, right now we need to do some more work, because we have now a null model. We have an idea of a new way that these forces might be being generated. And now we need to go into experiments where we stop the cell from moving through the tissue using a mutant, right? Or we can actually change the properties of the tail bud cells using some drugs. And so that's what we're doing right now. But I think it's interesting to say, hey, look, these physical forces all by themselves, without any sort of additional patterning or anything, could give rise to something that's really important for congenital heart defects. That's sort of the point. And it's really important actually for this result that let me just go back one slide. It's actually really important for this result that these tail bud tissue are fluid-like. They have to be fluid-like, but not too fluid-like. They have to be viscoelastic, like we talked about for this effect to really work well. Okay, so that's kind of interesting. The material properties of the cells surrounding this organ could be really important for this left-right patterning that's really important for us. Or maybe it's not, but we'll do some more science and find out. Okay. So then another story that I want to tell you about is sort of thinking about using some of these tools to understand features of cancer. And so we're doing these experiments both on primary tumors from patients with breast and cervical cancer and also on these breast cancer cell lines that I told you about. So these are not taken from human patients, they're just cultured in the lab, and these are taken from human patients. And so there's a lot of people working in the case group that are doing the experiments with whom we're collaborating. Okay, and so one of the things that our collaborators can do is they can take these tumor samples and they can take a large fraction of all of the cells in one of those tumor samples from a human patient, and they can send all of those cells through what basically is a light tweezer. So they can stretch the cells, and they know how hard they're stretching the cells, and they can look at the compliance of the cells, and they can basically see how stretchy the cells, how soft the cells are. And that's actually one of the parameters in our model that I showed you, this vertex-like model, is how much the cells want to maintain a sort of a spherical shape, okay? And so they get data that shows basically, so this is how much the cells stretch, and this is, again, measuring lots of cells, and that's important because cancer tumors are very heterogeneous. They have lots of different cell types within them, and so you want to measure a population of them. And so you can see that if I look at how stretched, this is the relative deformation, that's how stretched they get under a constant force, you see that there is a control that's a little bit less stretched on average, and the cancer cells are softer. And you can see that maybe you can see a little bit that they're also a little wider, that there's a more heterogeneity in the cancer tissue than in the normal tissue. And that's even more apparent in the cervical cancer case. So what we can do is we can sort of take distributions of cell stiffnesses like this, and we can translate it into our vertex model, and we can ask, does anything about sort of the interactions between cells within a cancer tumor either change the mechanical properties of the cancer tumor in a negative way, in a way that makes it more dangerous or more invasive, with the eventual goal of trying to have a mechanical marker for cancer invasiveness? Because actually, I mean, one of the largest problems with sort of developing treatments for cancer is that we really don't have a great system for telling how dangerous a cancer is. Sort of cancer staging is notoriously difficult. Pathologists get it about 80% right, okay? And so you'd like to have better ways of deciding how dangerous a cancer tumor is, and we think that mechanical phenotyping like this might be a way to do it. So that's the eventual goal. Okay, and so we take these distributions and then we fit them. And what we can do is within the framework of these models, we can basically now put a heterogeneous distribution of cell stiffnesses and ask, how does this group of cells self-organize, and what's the properties of it? Is it a solid? Is it a fluid? Okay, and so one of the really excellent postdoctoral associates in my group realized, so this is a picture showing how soft a cell is in our model. And again, we took this distribution of stiffnesses from the cancer tumors, and you see that if you actually sort of split it and you say, well, let's just guess that basically if a cell has a P0 that's greater than the special value, that it's soft, and if it's less than that special value, it's solid. So it's a hypothesis because I showed you data that was for a system of all the cells that were the same, right? I showed you that this was true if all the cells were the same, but you say, well, let's just see what happens if we assume that it's true on a cell-by-cell basis too. Then what you can see is you can ask, well, when does this thing transition from solid to fluid? Like, what has to happen? And so it turns out that the important parameter here is the fraction of cells that are basically solid, right? So if their value is less than this special number, then they're sort of solid-like. And you can basically see that if I try to understand how rigid a tumor is with cells of a given type, if I plot things as a fraction of just the rigid cells, then it all collapses. So on this side, basically there's no tumor rigidity, which means like it's a fluid, and then it becomes a stronger and stronger solid. And the interesting thing here is that rigidity transition here happens at less than 50%. And so this is interesting to us because like one of the questions in cancer sort of biology is that it's known that individual cancer cells are soft, but the tumor as a whole is still rigid. And it's actually important that it's rigid because that actually leads to it being able to pull on the cells around it and stiffen further. So it's sort of an important thing. And so what we find is that these things can rigidify, these sort of cell-cell interactions. They can generate a rigid tumor even when more than 50% of the cells are soft. So it's kind of cool. For those of you in the know, it's a percolation transition, okay? And then you can actually look and ask, well, are there any interesting patterns that form in the dynamics of systems like this? Because you also want to know, does this heterogeneity set up anything dangerous for allowing cells to exit a tumor? And it turns out that in the systems that are heterogeneous like this, because there's a rigid backbone of cells, the soft cells actually form these sort of streams, sort of quasi-once-dimensional streams that can sort of exit the tumor. And those softer cells are thought to be sort of the more dangerous ones once they get out into your body. So it's sort of like, unfortunately, if you have a heterogeneous cancer tumor, it spontaneously develops the ability to send out these multicellular streams, and it's just a mechanical effect. Of course, there are also biological effects that allow these things to leave in streams, but it's kind of interesting that it can happen just physically. So I'll show you a picture of what happens in one of our simulations just to visualize it better, because these things are a little hard to visualize. If you start out with a patch of soft cells in the middle, and I'm not showing you rigid cells, but there's rigid cells all around this one. And so if you just allow the cells to move around a little bit, what you see is this sort of thing develops a spontaneous stream that sort of streams out in like a one-dimensional way. And that actually is what's seen in sort of pathology reports of sort of tumors. This is a well-known phenomena. Okay. So then I want to ask one more question, which is another question that's important for development, and also actually it's also important for cancer. So the idea is that in development, it's really important for your cells to sort of develop compartments. Those compartments form things like organs, right? And so it's really important to segregate different cell types from one another. And in cancer, there's a breakdown of this sort of segregation of compartmentalization, and it's one way of thinking about it. Okay, and so we want to try to understand how do tissues maintain boundaries. And so this is a picture where you can see that there happens to be a very wiggly boundary, but there's a lot of cells actually go to a lot of trouble to develop sort of these interfaces that are called heterotypic interfaces, which means you have two cell types that know that the other cell type is a different cell type, and they change the mechanical properties along this interface between two cell types. And so the question sort of for us is why, from a physics perspective, why do they go to so much trouble to generate these heterotypic contacts, to change their mechanical properties along this interface between them? Is it sort of necessary? Okay, and so if we're trying to understand the properties of these interfaces, what we can do in our model is we can just say, okay, let's see what happens if I have one cell type over here and one cell type over here, and I say wherever they touch, there's an extra tension in the model. There's a difference in the physical force along that interface. And you can ask to see what happens there. And so you can see that if sort of this thing gets compressed, that this is one cell type here, and this is a different cell type in the bulk. And this is just a type of experiment. Actually, people really do this experiment. This is called a parallel plate compression experiment, and biologists actually do it to measure tissue surface tension. But if you do it in our simulation, you can see that you can sort of measure how hard it is to compress these plates, and that's a measure of how stiff the interface is. So they do that. And what you find is that, I'm not going to explain all the details, but you find that there's sort of a sort of equilibrium force, and it's really kind of soft. It's a pretty soft mechanical interface. Another thing you can think about if you're trying to understand how stiff an interface is, well, how much does it fluctuate, right? Because if I have a very stiff interface, it should not fluctuate very much at all. And if I have a very weak interface, it should fluctuate a lot. And you can actually use the tools of statistical physics, and in equilibrium, statistical physics tells you that these two things should give you the same answer. If you look at fluctuations, you can map that on to this parallel plate compression experiment. So the mechanical measurement and the fluctuation measurement should match. Okay, and I want to point out that it's sort of, in this simulation and actually also in experiments, it's surprising already just looking at it how sharp this interface is, because really it's very much smaller than a typical cell diameter in these types of simulations, and it's very different from what you see sort of, I guess, at some other types of interfaces in fluids, even between two immiscible fluids. Okay, and so we can actually try to quantify that. And again, this is a plot that I'm not going to tell you all the details of, but I'm going to tell you this is the result of what you get. This is the sort of what you get if you try to measure how big that interface is if you have sort of one of these fluctuation measurements. So right now you can just focus on that green line, okay? And then I'm going to show you what the prediction is for how big this width should be based on that mechanical measurement, okay? And so if I show you that, that's up here, okay? And so this is a logarithmic plot, which means the difference between this and this is an order of magnitude. So what this suggests is interfaces that have this property, they are very, very soft, but they are extremely sharp in a way that's not consistent with our usual understanding of statistical mechanics. For those of you in the know, it turns out that what saves you, so this is really true, it's a cool case of where the surface, the fluctuations are different, but what saves you is the nature of the interactions between cells is topological instead of geometric, okay? And so it turns out that for biology and for us compartmentalization, these heterotypic contacts are way better than you would expect at making sharp interfaces. And so they're really, really good at getting compartmentalization, much better than you would expect from statistical mechanics. And then you can do sort of simulations, and you can see that when you add these heterotypic contacts, you do a really good job of generating these little islands of one cell type in another. And in the absence of those heterotypic contacts, they don't segregate. Even if you have two different cell types, but they don't have the special interfacial tension that allows them to be different at the interface, in these models, they don't segregate at all. So you kind of need this to get compartmentalization is the idea. Okay. And so there's a lot of other questions that we're thinking about with this model. Like, for example, I didn't tell you very much today about cell division and cell death, and that's because we still don't deeply understand it, but we're thinking about it right now. What you do know is it does tend to fluidize a tissue, but it's like really remarkably good at fluidizing a tissue, and we don't deeply understand why yet. At least I don't understand why it's so good, and so it's something we're thinking about. But you can see that if I have cell divisions in a strip like this, I can allow the strip to grow, and that's going to be an important component of these wound healing experiments. And there's a balance between forces at the edges and sort of the fluidity of the tissue inside. And so that's something that we're thinking about. Another really interesting behavior that's important in biology is collective cell migration, right? Where you have groups of cells that all move in the same direction to get to a new place within, say, an embryo. And one of the things that we're sort of trying to understand is what type of local alignment rules allows you to get sort of a collective flocking behavior. So what you're seeing in this movie is these different colors tell you sort of how aligned a cell's motion is. So it's sort of the colors tell you the direction in which the cell's moving. And you can see that there's little patches of particles that sort of move together for a while and then they separate again. And this is a sort of what I would call a flocking fluid. So it means that cells are still fluid-like, meaning they're changing their neighbors, but yet there's patches that have a collective direction that they like to move. And these sorts of pictures actually show up in some types of some of the pathologies that go with cancer. So if you take a normal epithelial cell type, like a normal cell type like for your skin, and you basically add to it a factor that makes it turn into a cancer tissue, it will generate patterns that look really similar to this because we think it's interfering with the ability of cells to align. And so another direction of this work is to try to understand how alignment of cells moving interacts with the mechanical properties of the tissue. Cool. Okay. So in summary, the tissues in your body can be thought of as a living material. That's sort of what I'd like to, I hope I may have convinced you of. And I want to say that I think sometimes it's useful for the tissues to be fluid-like. For example, in early embryonic development or when you're closing a wound. And sometimes it's really important for them to be solid-like, not only because you have to walk around and support shear stresses, of course, but also because during development, these animals often use buckling, like when you take a piece of paper and compress it and it generates a crease, to generate structures. And so there's this really beautiful interplay between fluid-like behavior and solid-like behavior in embryonic development. And I think it could be a motif that can help us to think more about the molecular mechanisms that are involved in some of these processes. The physics of glasses is a useful starting point for thinking about transitions between these two phases. We could never have done this work without all of the work that's been done already to really deeply understand some of the properties of glass transitions. We co-opted a lot of it to do this work. we can use simple models for these materials to make accurate predictions about tissue behavior. In other words, biologists are right now using these sort of very physicsy models to help them understand the mechanics of their tissues. And so we think that we can help them develop better null models for some of their systems using these physics principles. And we can use these insights to understand disease, but of course there's a lot more to do and the future is exciting. And before I conclude, I want to say that, science is something which is extremely collaborative. And so I want to show you all of the people who did this work from all over the world. These are people in my group here and people both sort of in Europe and people in the United States. And that, I think it's amazing how hard these folks work in order to do this science. They don't get paid very well, but they do it because they love it. And so I just wanted to show their pictures. And I wanted to also sort of say that these people could only do this work because they're funded to do this work, because people value science, right? And so in particular, as you heard, the Simons Foundation has done a lot to allow us to do these kind of things. And some of it's a little bit, out there. And so the fact that they're willing to allow us to use these physics models to learn about biology is a really nice thing. It allows us to do new sorts of things. And so that's much appreciated. And thank you for your attention and also for braving the snow.